She's beauty, she's grace, and she can be all in your face. The punchy personality that some wines have, like those big Barossa Shiraz, come from a little thing called tannin. Tannin in wine can come from a variety of sources such as stems, seeds, skins, and sometimes fermentation vessels like oak barrels. It can impart a bitter or astringent taste into wine, and keeping these levels balanced is critical to wine quality. Tannin levels are influenced by fermentation style. Red wines with longer skin contact and oak maturation have higher tannin levels and a richer phenolic profile. These tannins benefit aging, softening over time as they polymerize and reduce the wine's initial sharpness. In grapes, tannins originate from phenylalanine, an amino acid that feeds into the phenylpropanoid pathway to make flavonoid intermediates such as chalcone and naringenin. These are converted into flavonols like catechin and epicatechin. These can then join together to create tannins that influence a wine's mouthfeel, taste, and astringency. The Australian wine industry is exploring better ways to control tannin levels through breeding, but traditional crossbreeding methods like sexual and asexual reproduction are slow and unpredictable as multiple genes regulate tannin traits. More recently, a new method called CRISPR, also known as clustered regularly interspaced short palindromic repeats, has been researched. This technique allows specific genes to be edited while maintaining varietal identity, complying with laws and regulations that many wine regions have in place. It's also much faster, achieving results in years rather than decades.
Most grapevine varieties are self-pollinating plants as they possess hermaphroditic flowers. Male flower organs, the stamen and anther, are positioned just above the female parts, allowing pollen to easily fertilize the ovaries. Once pollen lands on the female part of the flower, fertilization occurs, inducing seed and berry development. The resulting seed can germinate in favorable conditions and produce a new cultivar with the mix of both parents' traits. In the past, accidental crossbreeding has had major impacts on the wine industry. In the 17th century, viticulturalists in Bordeaux cultivated the progeny of Cabernet Franc and Sauvignon Blanc. The new cultivar, Cabernet Sauvignon, had unique characteristics perfect for the region and is now the second most grown grape variety in the world, produced on every winemaking continent. Viticulturalists are also using intentional hybrid cultivars through human intervention in the pollination process. This involves introducing pollen from one cultivar to the female organs of another. To prevent self-pollination, breeders remove the male parts from the receiving flower before inoculating it with the desired pollen. An example of this is the crossbreeding of Pinot Noir and Cinsault, which produced Pinotage, a variety perfectly adapted to South Africa's climate.
CRISPR is one of the most exciting breakthroughs in modern science. It's a highly precise tool for editing DNA adapted from a natural defense system in bacteria. In nature, bacteria use CRISPR to recognize viral DNA and cut it apart, protecting themselves from infection. This mechanism inspired the revolutionary gene editing technology. In the lab, CRISPR works with two key components: the guide RNA, which acts like a GPS, directing the system to the exact spot in the genome that needs changing, and the Cas9 enzyme, which acts like molecular scissors to cut the DNA at that point. Once the cut is made, scientists can delete, repair, or insert new DNA sequences, allowing targeted genetic changes, whether removing a faulty gene, correcting a mutation, or introducing a new function.
CRISPR stands out for its efficiency, speed, and accuracy. Unlike older methods that were slow, costly, and unpredictable, CRISPR is faster, more affordable, and highly precise. The guide RNA can be reprogrammed to target almost any sequence, making it extremely versatile. This adaptability is transforming multiple fields from medicine to agriculture, where it's used to develop more resilient crops. Although CRISPR hasn't yet been widely used in viticulture, its potential is enormous. Red grape varieties could benefit from fine-tuning genes controlling tannin biosynthesis. Tannins are central to wine quality, shaping structure, mouthfeel, colour stability, and aging potential. With CRISPR, scientists could enhance these traits more precisely, improving consistency and quality in wines such as Shiraz or Cabernet Sauvignon. Beyond wine quality, CRISPR could strengthen grapevines themselves. By editing genes linked to disease resistance, it may reduce vulnerability to fungal infections like powdery mildew or botrytis. Targeting genes that regulate drought tolerance and water use efficiency could also help vines adapt to harsher climates and lower rainfall conditions, making vineyards more sustainable.
Our team interviewed Adelaide University PhD student Jacob Long, who specialises in gene editing in grapevines. He explained that CRISPR is a promising DNA-free gene editing tool that enables targeted heritable changes without introducing foreign DNA, making it suitable for large-scale commercial use. Using genetic mapping, researchers can identify and target specific gene responsible for desired traits such as tannin content or disease resistance. Jacob explained that reducing tannin through CRISPR is relatively straightforward, as it involves disabling genes involved in tannin biosynthesis. However, increasing tannin production is more complex. DNA-free CRISPR editing only removes or disables genes, so to increase tannin, scientists must need to locate and remove a tannin suppressor gene, something yet to be discovered. He also discussed how CRISPR is being used globally in agriculture, with examples including climate-resilient crops and high GABA tomatoes. In Australia, DNA-free CRISPR editing does not classify vines as genetically modified organisms under current regulations. This allows new cultivars to retain their varietal status and avoids negative consumer perceptions associated with GMOs. In contrast, European laws require even DNA-free edited produce to be labelled as genetically modified. Jacob also noted that even after a successful CRISPR edit, the new cultivar must undergo 10 to 15 years of biosecurity testing to ensure it imposes no risk to vineyards or the industry. He explained that CRISPR can improve disease resistance, not by adding resistance genes, but by removing susceptibility genes. This has already shown promise in trials reducing downy mildew infection in varieties such as Chardonnay.
Our team also researched a CSIRO case study by Dr. Paul Boss and Dr. Christine Botcher. The research used DNA-free gene editing in grapevines to address agricultural challenges like climate change and downy mildew. They successfully targeted susceptibility genes VVI-DMR61 and VVI-DMR62, linked to grapevine downy mildew vulnerability, and regenerated edited plants of 4 varieties, Chardonnay, Shiraz, Sauvignon Blanc and Cabernet Sauvignon, within months. The edited vines grew normally, and the fungal challenge assays showed reduced susceptibility to downy mildew. The study also revealed a link between VVI-DMR62 and jasmonic acid signaling, suggesting a broader role in plant defense. Dr. Boss and Dr. Botcher's work demonstrated that CRISPR can be used effectively in a DNA-free, non-transgenic way, meaning the resulting vines are not considered GMOs under current Australian regulations. Their work provides a proof of concept for how gene editing can help the wine industry face key challenges and offers hope for future research.
CRISPR offers a groundbreaking way to influence tannin without altering varietal identity. By specifically targeting genes that influence tannin production, it enables faster, more controlled improvements than traditional branding. As research advances, CRISPR could help Australia's wine industry adapt to change.